Imagine a child born with a really severe neurodevelopmental disorder. The condition is called GPT-2 deficiency.
Right.
Their brain is struggling to develop. And if you're just looking at this from the outside, you might naturally assume there's a fundamentally broken brain cell at fault, like a missing piece of anatomy.
Yeah, that would be the logical assumption.
But the root cause isn't a missing cell at all. It's actually a microscopic lock clamped onto their DNA. And it's there simply because of a missing metabolic key.
It's a tragic situation, but mechanically, it's just absolutely fascinating because the actual instructions for healthy brain development, they're perfectly intact in that child's DNA.
Wow, really?
Yeah, but the cell has essentially padlocked those instructions completely shut. And the specific molecule, like the actual chemical key needed to pop that padlock open, is just gone.
Which brings us to the core mission of today's deep dive. We have this massive stack of data from a groundbreaking 2026 paper in the journal Science.
It's a brilliant paper.
It really is. And we're looking at how our cells regulate a crucial molecule called α-ketoglutarate, or IKG. So our goal for you listening today is to unpack how researchers literally hacked a survival mechanism from pond scum to build a microscopic spy.
Right, a literal glowing spy.
Exactly. And how that spy exposed this hidden highway inside our cells, and how this wild discovery might actually offer a cure for devastating genetic diseases like GPP2 deficiency.
It's a journey that directly connects the food our cells burn for energy to the physical structure of our genetics. I mean, we are basically rewriting the textbook on how metabolism talks to DNA.
Okay, let's unpack this from the ground up. We need to start with the star of the show here, alpha-ketoglutarate, IKG. I vaguely remember this from high school biology as part of the Krebs cycle, like the engine in our mitochondria that makes energy.
Yeah, exactly, the Krebs cycle.
So why is an energy molecule acting like a key to a genetic padlock?
Well, because it has this massive moonlighting gig. So yes, ACO-KG is a standard metabolic intermediate, it's a fuel source. But it is also the absolute essential fuel for a very specific class of enzymes called dioxygenases.
Oxygenases.
Okay. And we're talking primarily here about TET enzymes and KDM enzymes. So these enzymes live inside the nucleus, the cell's vault, and their sole purpose is to demethylate your DNA and your histones.
Okay, let me just stop you right there because we're hitting the heavy biochemistry pretty early.
Oh.
So histones, demethylation, let's try to visualize this for everyone.
Good idea.
Think of your DNA as this massive instruction manual for building and running your body.
Yeah.
But it's millions of pages long, right? So to actually fit inside the nucleus, it gets wrapped tightly around these little protein spools.
Right, and those spools are the histones.
Okay, and that wrapping is highly regulated, right? So methylation, these chemical tags that cells attach to the DNA and the histones, is essentially like taking a thick black marker and just crossing out certain sentences so the cell can't read them anymore.
That's a perfect analogy, yeah.
So the AKG we're talking about, that's the chemical eraser.
Exactly. It literally powers those TET and KDM enzymes to just scrub away the black marker so the genes can be read again.
Oh, wow.
Yeah. TE and KDM are the erasers, but they are completely useless without AKG to power them. So if the nucleus runs out of AKG, well, the black marker takes over.
The DNA in those histone schools just get permanently locked down.
Right. They become hypermethylated. Genes that should be actively driving, say, brain development get permanently silenced.
Which sounds completely disastrous. So my immediate question is, why was this so hard for scientists to figure out? I mean, if AKG is the erasure fuel, why couldn't researchers just, stick a microscopic probe into the nucleus and see when it runs out?
Because of the sheer chaos inside a human cell. I mean, intracellular AKG isn't just sitting in one neat little puddle somewhere.
Right.
It's regulated by this crazy network of over 100 different enzymes and transporters. It's being created and destroyed constantly. And the traditional tools we use to measure chemicals in distinct cellular rooms, like the mitochondria, they just don't work for the nucleus.
Why not?
Because the nuclear membrane is highly porous, chemical pools are constantly washing in and out. So researchers needed a way to spy exclusively on the AKG that was inside the nucleus and just ignore all the noise from the rest of the cell.
And this is where the pond scum enters the chat. I love this part so much. To solve a human brain problem, these highly advanced researchers turned to cyanobacteria.
Yeah, cyanobacteria are ancient, and they've evolved this incredible survival mechanism for when they get well, hangry, specifically when they are starved of nitrogen.
Okay.
When nitrogen levels drop in their environment, ACG physically builds up inside the bacteria. And they have this transcription factor, a protein whose job is to turn genes on or off called NTCA. And NTCA is exquisitely sensitive to ACG.
So it's basically a bacterial alarm system.
It really is. When NTCA senses that massive spike in ACG, it physically binds to the bacteria's DNA and activates a whole suite of nitrogen-scavenging genes just to keep the organism alive.
So the researchers looked at this and thought, well, what if we just steal this bacterial alarm system, drop it into a human cell, and somehow rig it to glow green whenever ACG is present?
That was exactly the dream. But actually building this chimeric biosensor required agonizing granular engineering. You can't just drop a bacterial protein into a human cell and expect it to do anything useful.
Right. It would probably just get destroyed.
Exactly. So they took the NTCA protein, and first they had to fuse it to a human transactivation domain called VP64.
Wait, a transactivation domain? Explain that like I'm 5.
Okay. Think of NTCA as the hands that physically grab the DNA. But VP64 is the megaphone that shouts at the human cell to start transcribing a gene. You need both.
Okay, got it.
But here was the first major hurdle. The human cell recognized this weird VP64 megaphone and kept actively kicking it out of the nucleus.
Like a microscopic bouncer throwing them out of the club.
Yeah. That's exactly what it was doing. They actually had to analyze the VP64 peptide string and surgically remove these things called cryptic nuclear export sequences.
The hidden tags that were telling the cell to eject it.
Right. And then, to make sure it stayed put, they attached 2 SV40 large T-antigen nuclear localization signals, or NLS.
Which is basically forging a VIP pass to get past the bouncer and stay in the vault?
Yeah, it completely forces the biosensor to stay strictly inside the nucleus. But then finally, they had to build the visual alarm itself, so they designed a synthetic promoter using an ACG response element.
A genetic tripwire.
Exactly, derived from cyanobacterial genomes, specifically the GLNA or GLNN genes. And they placed that tripwire right next to a gene for green fluorescent protein, or GFP.
The glow stick. So let's trace the actual mechanism here. The NTCA is sitting in the human nucleus as a homodimer, meaning it's 2 identical protein halves kind of hugging each other.
Right, a homodimer.
So when nuclear ACG levels rise, the ACG binds to this NTCA hug. And the affinity here is wild. I read that the researchers calculated it binds with a dissociation constant, a KD, of 2.0 millimolar.
Which is the total Goldilocks zone.
How so?
Because that grip strength of 2.0 millimolar perfectly aligns with the natural concentration of ACG in a human cell, which hovers around 1.35 millimolar.
Oh, I see. So if it were too sensitive, it would just glow green all the time.
Right. And if it were too weak, it would literally never glow, even during a massive spike.
Okay, so it binds the ACG, the NTCA protein physically changes its shape, its quote-unquote hands grip, the major grooves of the DNA, the VP64 megaphone shouts, and boom, the cell glows green.
That's the mechanism.
But hold on, I'm finding a hole in this logic here. You just said earlier that ACG is everywhere. It's in the mitochondria, it's in the fluid of the cytosol. If this bacterial sensor is just sitting there in a porous nucleus, how do these researchers know that green glow actually means the nucleus is full of ACG, and it's not just reacting to a general flood in the whole cell?
That is the exact critique a peer reviewer would have. And, the researchers completely anticipated it. They validated their tool with this brilliant experiment involving 2 rival enzymes, BCET1 and BC2.
Okay, what do they do?
They are enzymes that basically eat ACG, but they live in entirely different cellular neighborhoods. BCD1 lives in the nucleocytosolic compartment, so basically right outside the nucleus door.
Right.
But BC2 lives strictly trapped inside the mitochondria.
Oh, wow, I see where they went with this.
Yeah. When they genetically deleted BCT1, effectively destroying the enzyme that eats ACG near the nucleus, the biosensor lit up like a Christmas tree. Nuclear ACG completely spiked. But when they measured the total ACG in the whole cell, it barely moved.
Because the spike was highly localized to the vault.
Exactly. And they flipped it.
Yeah.
When they deleted BCT2 in the mitochondria, the total cell ACG skyrocketed. The whole cell was flooded. But the biosensor in the nucleus, it stayed completely dark.
That is incredible.
It definitively proved the biosensor is compartment-specific. It only reports on the pool of ACG directly available to the DNA.
That is airtight logic. Okay, we've covered a lot of ground in our first 5 minutes. Let's do a quick grounding summary for you listening. If you're taking notes, here are the 2 big takeaways so far. First, ACG acts as a crucial epigenetic eraser in the nucleus, fueling the enzymes that keep our DNA from getting permanently locked away by methylation. And second, to actually measure this, researchers engineered a glowing ACG biosensor from starving cyanobacteria, slapped a VIP pass on it to keep it in the human nucleus, and proved it only spies on local nuclear ACG.
Perfect summary.
And this brings up a great question for you to think about. How crucial is it that different rooms in your own cellular house, the mitochondria, the nucleus, have their own separate, tightly controlled chemical stockpiles, rather than just sharing everything equally?
Honestly, it's the difference between a finely tuned machine and total biological chaos.
Which pushes us into the next massive phase of this paper. They have this incredible glowing biosensor working. Now they want to use it to answer the real question. Who is actually delivering these ACG erasers to the nucleus in the first place? Like, how does it get there?
Right. To map that highway, they set up a forward genetic screen using CRISPR-Cas9 on a line of U251 glioblastoma cells.
Glioblastoma, so brain cancer cells.
Yes, very robust for this kind of testing. They built a custom library targeting 127 specific metabolic genes. So these are genes responsible for synthesizing, transporting, or destroying ACG.
A forward genetic screen is just exhausting lab work. For anyone who hasn't done bench science, they essentially use CRISPR to break one of those 127 genes in thousands of different cells one by one, and then just watch the glowing biosensor to see what happened.
It is incredibly tedious, but it's powerful. They used fluorescence-activated cell sorting, literally a machine that sorts cells one by one based on how much light they emit. Yeah, so they isolated the top 10% brightest cells, meaning the nucleus was hoarding ACG, and the bottom 10% dimmest cells where the nucleus was completely starving.
But wait, in any screen like that, you're going to get false positives, right? Like a cell might glow bright green just because it's sick or dying and naturally autofluorescing, not because it actually has high Kg.
And this is where their granular engineering pays off yet again. Alongside the functional biosensor, they ran a parallel screen using an inactive mutant biosensor called NTCR90E.
Let's break down that acronym, R90E. Does that mean they swapped out an arginine amino acid for a glutamate amino acid at position 90 on the protein chain?
You got it perfectly.
Yeah.
And that single tiny amino acid swap completely breaks the protein's hands. It physically cannot bind APG anymore.
Oh, that's so smart.
Right. So if a CRISPR mutation caused a cell with this broken sensor to glow green, they knew immediately it was a false positive. It was just an artifact, a dying cell, not a true spike in AKG. They used this mutant to perfectly filter the noise out of their data.
I mean, that is just incredibly smart experimental design. So they run the screen, they filter the noise, and the data spits out the regulators, the actual molecular network, controlling the epigenetic eraser. So let's break down the hits. What did they find?
The screen revealed 4 dominant genes. 2 of them were suppressors, meaning when you delete them, nuclear AKG goes up. Those were DLD and the one we mentioned earlier, BCAT1.
Right, the one outside the nucleus.
Exactly. But more importantly, there were 2 suppliers. When you delete these, the nucleus goes totally dark. It starves. And these genes were GPT2 and SLC25A11.
Okay, I want to give everyone listening a mnemonic device here to remember these 2 vital suppliers, because they are the absolute key to the whole disease model later on. Think of the letters GST. GPT2 synthesizes, SLC25A11 transports. GST.
That's a great way to remember it.
Let's build out a mechanical analogy to visualize this hidden highway. So GPT2 is a manufacturing plant operating deep inside the mitochondria. Its entire job is to take raw materials and build the AKG. But the AKG is stuck inside the factory walls. It can't just leave.
Right, because the mitochondria has a double membrane that AKG cannot just passively float through.
Exactly, and that's where SLC25A11 comes in. It's the transport truck. Specifically, it's a key part of what biochemists call the malate aspartate shuttle. And this isn't just a passive pipe, right? It's an active pump, physically burning energy to shove the AKG cargo out across the mitochondrial border.
Yeah, it pushes it out. And once it's pumped into the fluid of the cytosol, it diffuses freely through the nuclear pores, entering the vault where the DNA is kept, ready to fuel those erasers.
And meanwhile, BCKT1, that suppressor we mentioned earlier, is acting like a rival factory, sitting right outside the vault, constantly eating up any excess AKG and turning it into glutamate, just ensuring the nucleus doesn't get flooded.
It's an entire, tightly regulated microeconomy.
It really is. But I know scientists don't just trust a genetic screen on its own. How did they prove definitively that GPT2 is the actual factory making the specific AKG that ends up in the nucleus?
They proved it using heavy isotope tracing.
Oh, tracing. Okay.
Yeah, so they fed the cells a heavy, non-radioactive form of nitrogen, specifically alpha-15 and glutamine. By feeding the cells this heavy nitrogen, they essentially slapped a GPS tracker on the raw materials.
That's brilliant.
So they could literally watch under mass spectrometry as GPT-2 took that heavy nitrogen and directly synthesized the molecules that would eventually become the nuclear EPG pool.
Okay, so we have the highway perfectly mapped. GPT-2 synthesizes, SLC25A11 transports. But what does this actually mean for the DNA? Like, what happens if you blow up the highway?
It's catastrophic. When the researchers deleted GPT-2 or SLC25A11, the nucleus starved. And without the EPG fuel, those TET and KDM erasers just ground to a halt. The black marker took over completely.
The hypermethylation.
Yes. The researchers observed massive hypermethylation of specific histone marks, spots on the spool where DNA gets locked down, specifically marks known as H3K4Me3 and H3K9E3.
So the DNA was getting permanently padlocked, but they could fix it, right? Because they know the missing ingredient.
Yes, exactly. They supplemented these starving cells with DMIG. Now, regular EPG doesn't cross cell membranes very well, so DMIG is a special esterified form.
Esterified, meaning they chemically coated it in fat so it slips right through the cell's outer membrane.
Precisely. It bypasses the broken transport system entirely, floods into the cell, and completely rescues the hypermethylation. The erasers turned back on, the padlocks opened.
Wow. Okay, we're about 10 minutes in. Time for our second quick summary for you listening to keep all these moving parts straight. First, by using a tedious CRISPR screen and an incredibly clever broken biosensor to filter out false positives, the researchers mapped a dedicated mitochondrion-to-nucleus highway. Second, remember our GST mnemonic. GPT-2 makes the EPG in the mitochondria, SLC25A11 exports it via an active shuttle, and the nucleus relies entirely on this delivery system to keep DNA methylation tags in check.
Spot on.
And here is your question to ponder before we move to the final act. If the mitochondria are widely known just as the powerhouses of the cell, generating energy, could their specific metabolic output actually be dictating which of your genes are physically turned on or off?
Which perfectly sets up the final and frankly most mind-blowing section of this research, because everything we've talked about so far is in vitro cell biology, cells in a dish.
Right.
The ultimate test is in vivo mammalian physiology. Does this microscopic highway actually matter in a living, breathing creature?
And it matters immensely. Let's circle all the way back to the human medical case we opened the show with. GPT-2 deficiency. In humans, inactivating mutations in the GPT-2 gene cause a severe inborn error of metabolism.
It does.
It results in devastating neurological defects, severe intellectual disability, and developmental delays.
And based on what we just learned, we know exactly why. The manufacturing plant in their mitochondria is broken from birth. To study this in a living system, the researchers turned to a mouse model, specifically GP2 knockout mice.
Meaning mice genetically engineered from birth to lack the GPT-2 enzyme entirely. So what did the DNA in their brains actually look like?
It was profoundly dysregulated. The brain tissue of these knockout mice showed massive increases in both histone methylation, those H3K4Me3 and H3K9E3 marks we mentioned earlier, and global genome-wide DNA methylation.
Like everywhere.
Yes. Specifically, they saw massive enrichment of 5-methylcytosine, or 5-MyC. The epigenetic padlocks were clamped shut practically everywhere across the brain's genome.
But I recall a crucial, almost a bizarre detail from the data here. This hypermethylation was highly specific to the brain, right? When they checked the kidneys of these exact same knockout mice, the DNA was totally normal.
That's true.
Why would the brain be locked down, but the kidney is totally fine?
Because different tissues rely on entirely different metabolic pathways. The developing brain is uniquely, exquisitely dependent on the GPT-2 factory to supply its nuclear EPG. Kidney tissue evidently has alternate manufacturing plants, backup systems it can rely on.
I see.
But the brain does not. Without GPT-2, the brain's neurodevelopmental gene expression programs just completely derail.
So this leads to the million-dollar question. If you have a mouse with a genetic structural problem in its brain, its DNA is physically hypermethylated because it lacks a crucial gene, can you fix it just by giving it a metabolic supplement?
Well, they had to try. So they administered that same DMAG, the fat-coated cell-permeable supplement, to the living GP2 null mice.
And what happened?
It worked. The DMARG infiltrated the brain tissue and successfully restored normal chromatin methylation patterns. It erased the aberrant padlocks.
No way.
Yeah, but even more incredibly, it had a macrophysiological effect. It promoted actual physical fitness. These knockout mice usually struggle with stunted growth, but the daily supplement rescued their weight, bringing them much closer to the healthy wild-type mice.
I really need to pause here because the implication of this is just staggering. What you are saying is that a genetic, physical, structural problem in the brain, the hypermethylation of chromatin, was fundamentally uncoupled and fixed.
Yeah.
Not by gene therapy, not by CRISPRing the mouse's DNA, but simply by providing a missing metabolic drink.
That is the exact paradigm shift this paper represents. It proves that compartmentalized metabolism, where chemicals are and how they move, is a foundational determinant of the epigenetic state. The disease, GPT-2 deficiency, isn't just a generic broken cell problem.
Right.
The literal driver of the neurodevelopmental defects is the localized starvation of nuclear AKG.
Because the erasers run out of fuel, the DNA gets locked, and the brain can't develop.
And by identifying that highly specific mechanism, this research offers a tangible potential therapeutic strategy for patients. I mean, if we can find ways to safely and effectively supplement the missing metabolite in human brains, we might be able to unlock those genes and counteract the pathogenic mechanisms of the disorder without ever even touching the DNA itself.
That is just incredible. Let's do our final quick summary to lock this all in before we wrap up today's deep dive. First, the loss of the GP2 gene in living mouse models specifically starves the brain of AKG, causing massive DNA hypermethylation and severe developmental defects. Second, supplementing these living mice with DMAG actually restores the epigenome and dramatically improves their physical health and weight. And third, this proves that metabolic supplementation isn't just about giving a cell energy. It could be a highly viable, targeted treatment for genetic structural disorders.
It really illustrates why understanding the deep granular why and how of cellular mechanics is so absolutely vital. We had to go all the way down to a starving pond scum bacteria, build a synthetic biosensor, map a microscopic shuttle bus highway, just to understand that a devastating brain disease was actually a localized metabolic fuel shortage.
Which leaves us with a really profound, lingering question to close out this deep dive. We started by talking about a microscopic lock on the DNA, caused by a missing metabolic key. We now know that a simple metabolic intermediate like IKG can cross organelle boundaries, travel from the mitochondria into the nucleus, and literally unlock DNA in the brain.
It fundamentally connects what a cell consumes with how a cell is literally structured.
Yeah, exactly. So for you listening, I want you to really think about this as you go about your day. If a single molecule like IKG can potentially treat a severe neurodevelopmental disorder by rewriting epigenetic tags, what other dietary or metabolic molecules are silently shaping your epigenetics right now? Could the fuel you're giving your metabolism today literally be rewriting the physical structure of your DNA tomorrow? It's a wild thought. Keep mulling over that massive paradigm shift, and we'll catch you on the next deep dive.